And our next speaker is Vadigepalli from Thomas Jefferson University, and he's going to be talking to us about the convergence of single-cell assays, bioinformatics, and simulation to study tissue injury and repair.
Okay, thank you. Can you hear me okay? Yeah. Thank you, the organizers, for putting this together. It has been a fantastic learning experience so far, and I look forward to almost a day more left here. And thank you for inviting me to give an opportunity to share our experience, our learnings, and maybe some perspectives on how we can take some of the methods going forward. Rather than talk to you about the methods and so on, I thought I would try to present a certain conceptual framework of how we have been thinking about taking advantage of single-cell scale technologies, if you will, and why I feel that the operating word is convergence, how we have to put together multiple ways of perspectives of looking at the same problem and come up with the insights that any particular technology, one way of answering the question would not. And it seems intuitive, but I will actually make it very concrete and very specific as I go along and give you some vignettes on how this happens. And no disclosures. If you want to change this thing, talk to me later. None of this will happen, wouldn't have happened without the team of teams, if you will. It's truly networked science through which I have grown, operated, and learned, and done whatever science that we have been able to so far. And there are twice as many alumni that are not even represented here. And it goes without saying that NIH plays a big part in making all this go for a long time. There is no yes yet, Linda. I want to bring us back to this. We all know this a little bit, but I thought this would be a good starting point to agreed upon common ground, if you will, as a biology, as a way to discover. As we talk about atlases and characterization, we want to develop these atlases not because we want to do stamp collection and label and table and so on, but rather we want to reason, we want to apply, we want to create based on those. We want to do something else, synthesize with that information. And the single cell changes that game of the tabulation, game of that atlas building completely. This is just one example of, this could stand for gene expression, metabolomics. It doesn't matter how you think about it. The dimensionality of it, the resolution, the throughput is unprecedented. And yet, at the end of the day, if you think about it in a certain way, it's a completely new, but an atlas building tool, a characterization at a level that's not been available before. But how does this inform a conceptual way of looking at biology as we discover new things in biology? And as an example here, you can turn those big tables into interesting visualizations that are informative about cell type distributions, and so on and so forth. But what I would say is you actually have to put it together in a convergence framework, if you will, where we are talking about taking in vivo data of big data, small data, any data, knowledge sources, along with what I would say a statistical way of looking at these things, together with a dynamic modeling theoretical framework. Because these are fundamentally different perspectives that come up. None of them can truly give us the answer. They're just one window of what the mechanisms or pathways that we want to discover. That's because the questions are, the kind of questions, broadly speaking, are very different. The in vivo, it's about what is. It shows us what it is with the fantastical imaging tools that we saw today. And they're more... the statistical gives you probabilistic idea of what might be latent, buried in the data, the patterns that are in the data. But you really want to ask the question of what if, and particularly in the context of environmental health and drug discovery and so on, you can't really ask this what if questions with in vivo models. So we take it for granted that we use simulation modeling, but what I'm suggesting here is that you actually have to think of them in a very deliberate framework in which you expect to derive insights from a convergence. It doesn't mean that you're moving from one to another in a linear way. It's extremely flexible, and I'll illustrate multiple ways of entering this whole framework and getting to the middle as we go along. So I want you to keep this framework in mind, and I'll keep returning to this as we go along. And one of the first vignettes I'll give you is looking at what is the plasticity of molecular states within a subtype or two. So I think that was a question that probably Lisa posed earlier today about within a subtype, within that cloud, within that blob, what happens when you peek closer, right? And so, which is what we attempted to do a few years ago by looking at neurons in the brainstem that would make catecholamines versus the ones that would not. And we have ways to identify those two separately. And we use laser capture, so it's not dissociation technique. It is pre-drop-seq being useful and available for everybody. And so this is painful, one at a time, left with laser capture microdissection method. And we didn't do transcriptomics either, but we measured a heck of a lot of genes that are relevant to neuronal transmission, biosynthesis of neurotransmitters, channels, receptors, and what have you. And cutting to the chase, the idea is what we find is these so-called two fundamentally different cell types really organize themselves along a gradient of these molecular programs. So the archetypes of catecholaminergic cell and a non-catecholaminergic cell that you think of is pretty much an analog, flexible, plastic regulation that could be turned on and off in a particular way. And what I'm not showing here, and you can go get to the paper, is with a physiological perturbation, you can move this gradient around. And that's where the plasticity comes from. The underlying regulatory networks are way more flexible in taking the blob of within a subtype, within a cell type, and organizing it along some functional gradient, if you will. And if you need more catecholaminergic cells, then all it takes is a regulatory network pushing this whole molecular program in one direction and not the other. Yeah? So that tells us there's a different way to think about this, but then what shapes this? What shapes this distribution, right? I was talking about convergence. Where is the modeling coming into this whole picture? So we then basically looked at a couple of genes that are immediate early genes. And here we had perturbed the system in a physiological way by changing blood pressure. So we want to look at the evoked gene expression in those cells, not the homeostatically expressed ones. And we took the single cell data and said, what kinds of inputs, if you will, in this system, the signaling inputs, can fractionate it into these groups, right? Can we infer something about the inputs that these neurons are receiving? And for that, we formulated a fairly straightforward computational model of how certain immediate early genes are triggered downstream of known signaling pathways. And there are feedback loops and feed-forward loops, and you can actually intuit what might happen in this that easily. You would have to put a dynamic model together and simulate, and which is what we did. But rather than just look at one possibility, we said, what happens when the duration of signaling of various inputs that are coming in varies over a range, wide range? And so, of course, the outcome varies over a wide range, but you can take the extremes of it and then go, can we learn something about what those, is it random from here to the input space to the output space, if you will? Or is there some structure to this? Is there structure to this variation that we are seeing earlier? Is there any way to go back? Yeah. So is there structure to this variation that we see, right? That's what we want to try and understand with the modeling. And the final result, when we take all this variation data from simulation here, the what-if simulation, right, and plug that into decision trees and classification-based methods, what we see is that depending on the duration of, in this case, of how long the peak of a signaling activity is retained for specific signaling molecules, you get to a different parts of that variability, that blob, if you will. So if the signaling molecules are active for a very long duration, you get to the top part of that blob. If they're active for very little, you get to the one end of it, and any kind of a mixture gets you in a different part in that multidimensional space. So that means the plasticity that we see is potentially governed by the inputs that the cells are receiving in a functional tissue at any given point of the time. And again, remember, this is duration. So this is not about instantaneous activity. This is about history of what the cell has seen over a certain period that's being encoded into the variation that we see. So suddenly what that means is, from a snapshot single cell data, we can learn something about the temporal history of that cell if we were to use a physics-based dynamic model. And that makes it those data sets even more powerful than what they are as just a representation of what is at any given time. So that's the convergence insight, if you will, right? Learning about the history of these cells by putting all of these things together. And I would argue that you couldn't have really learned that from any individual aspect of this per se. You have to put it all together from multiple aspects. And we have done similar thing for a central circadian clock as well, where We had spatially tracked down where individual cells are in the suprachiasmatic nucleus of the mouse brain and put it together and developed a new circuit of the local paracrine microenvironment. I won't go into the details, but it follows a general idea of what I just showed. But we don't have to start from let's go collect single cell data and then infer some model and then see if it turns out to be true or what we can learn from it in a linear way. We could start with a model. Because we're not starting biology at zero, right? We already know quite a bit to formulate a what might be, the what if of the system that is of interest to us. And I'll illustrate that in a little more detail in the context of the liver. So liver injury and the liver response to injury, this is very well known for a long, long time now. And for about a century or so, we have developed animal models that really let us get to it in a very systematic, defined way. And what is fascinating is after removal of a portion of the liver, it comes back pretty close to where it started. And so this is what the surgeons rely on pretty much in every liver surgery resection that they're conducting and the live donor transplant, small size transplant. This is what enables all that. But how this actually works still remains a mystery for the most part. We have a long list of players and we have networks, hairballs, all this being drawn. But how it actually works, where is the control point, in what context, under which disease context, that still remains a mystery and keeps us in business for a while, I suppose. So we've been going at this from a convergent systems pathology angle for a little while, and I'll try to give you a couple of vignettes into this, both from a gene expression as well as from single cell live imaging point of view. So we want to start with a model this time. Put a formulation together of how multiple cell types might interact in a tissue that is responding to an injury. And it's not just cell types, it's cells in different functional states that they are traversing and transitioning in response to molecular cues. And these cells, depending on their state, they change the molecular milieu. So it's a multiscale back and forth molecular cellular interacting network that you can formulate based on all the review papers you want to read. Okay? The idea is, it's actually fairly straightforward to, I'm showing you a much simplified example where if you plug it in with the known kinetics of cell cycle, known kinetics of signaling molecules and so on, you can simulate, and this has a direct complement to the single cell states, because at any given point you can say what are the relative proportions of various cells in various states, and if you have a molecular way to characterize them with your single cell techniques, then you can test this model predictions and test all its what if scenarios, right? That's a general idea. And we are doing this obviously with a much more complicated model, and I'll cut to a very small aspect of it, which is we want to look at what are the control points due to the hepatic stellate cells? I think Vasily mentioned hepatic stellate cells in this presentation as well. So what is the transition balance between different states that the hepatic stellate cells can take, and what impact might that have on a regenerating system? So we can ask this question purely from a modeling point of view, and what we find is if you look at the relative proportion of those cells and you change the transition rates, the balance of the transition rates, the higher the balance is, so the higher the balance is going towards one state versus another, the whole system shifts. And as the system shifts, the regenerative response is suppressed. So it's almost as if this balance of these transition rates between functional states acts as a gain controller. So what governs those kinetic transitions between things? That would be the underlying signaling pathways, the receptors that they express to be able to respond to molecular signals one way or another. Because all the molecular signals that would drive towards one state or another are already present. It's just how the cell would choose to respond to one or another, right? So that balance is what governs the gain control of this. And so we went again into laser capture to go test this and see if we can find these proportions of 3 different states and how they might change. And biology always has a way to smile back at an engineer and say, nah, go back to your drawing board, right? So we find 4 states and not 3. So that sort of confuses the issue a little bit, because we accounted for 3 and the dynamics is dependent on a specific way those interact, and the underlying biology is telling us it's actually 4 states that you need to worry about, not 3. And so then if it is 4, then what possible ways can they interact? So we have to go back and start thinking about collecting time series data sets possibly and figuring out which of these kinds of models are compatible with the tissue injury response that we do observe. And switching over to the hepatocyte states, so we got to update that, but looking at the hepatocyte states, when we measured the hepatocytes, thankfully, at least in this case, that they seem to be in 3 fundamentally different states, which is what is expected. The cells that don't replicate, cells that are primed and responsive to cytokine signals, and cells that are in some stage of a cell cycle or another. And so if you were to take a disease model and look at the same thing, it turns out they're somewhere in the middle. There are all sorts of new transitory states that come into play. They're not in some other random state. The proportions don't shift. There are all these transition states suddenly that show up. That means we got to start updating our mental model of even the actual quantitative model of how we think about it. It's not about canalization and particular buckets that we put functional states into, but rather a flexible landscape that the cells are moving back and forth, and we have to think of a cell state as an analog, the cloud blob that we started with in the first place. And this abstraction may actually be hampering us in trying to understand this, which means we've got to go back to the dynamic model again, back to the drawing board, and fix it up and come back into this convergence thing, because the results from here and here, we can't put them together. They're not compatible the way they are right now. So that involves going back again and trying to figure out a new model. So I'll give you one other example, which is nothing to do with gene expression data, very little to do with it, so that you can see that this is not just about single cell gene expression data. And this is looking at calcium. Calcium as one of the master regulators of any number of functions in the liver, and we are interested in connecting calcium signals to the metabolic aspects and drug metabolism and energy metabolism. And what it is this is live imaging in the in vivo system, right, in the liver, and they have traveling waves that seem to mostly emerge from around central vein and travel, and we could actually resolve this. There are about 1,300 single cells that can be segmented out, which we have done manually, one of the graduates in the lab, and you can apply statistical analysis to figure out what are the possible ways the cells are influencing each other in these calcium patterns. And it turns out that the orientations of influence are all over the place. They're not going in one radially out from a central vein. So the statistics is telling us the pattern that you expect is not what is real in the system. So how does the traveling wave emerge then, when the influences are in any random direction between the cells? So we got to go back to the what if question again and ask that, right? And one of the things we had to do was we looked at a single-cell RNA-seq from Weizmann Institute that was available at the time, and we found that some of the calcium signaling pathway components that we're talking about are zonated. So if we take advantage of that information and initially ask the what-if question, what if the signaling pathways are not zonated? What happens? The experimental data shows a clear traveling wave, and if the cells don't talk to each other, you don't actually see a wave, so cell-cell gap junctions are crucial for this. And you don't see the wave the way you expect if they're randomly set up, even though cells can synchronize each other. So you actually need the zonated signaling pathways and cell-cell contacts, as well as connectivity that's randomly oriented in order to observe the traveling waves. But what does a random orientation give you? It turns out that it gives you robustness. You knock out certain connectivity, for example, in disease, maybe the gap junction expression goes down and some cells are not able to communicate with others in some random way, possibly. You take out 50% of it, you can still get a traveling wave, which means a tissue scale metabolic synchronization can still happen even if only 50% of the cells are able to communicate with each other through molecular exchange. So if there was a directional orientation, that would be cut out. Whereas a randomized orientation of interactions would actually help that cause. And that's what, so it's but of course the real system is 3D, not 2D. So we are going through some pain to get an extremely high resolution detail lobule reconstruction from 3D confocal imaging stacks. And we have some initial simulations where we can do it in 3D and start asking these kinds of questions of a 3D simulation. And almost the last slide here. The point is that we can actually start tracking 3D microdomains of peak calcium responses as traveling waves in 3 dimensions. So what is observed as a traveling wave in a 2D projection that we see is a microdomain that's in 3D that's moving around, that's synchronizing and organizing. That changes the way you think about the tissue completely. It's not about a 2-dimensional radially outward view in any which way. It's about synchronization of a whole host of cells in 3 dimensions. And so And there is no way you would have come up with any of these concepts if you're only looking at one or two of these ways of looking at this problem. The convergence is where the new insights that are emerging in this way, in a particular way, and they depend on all of these. So this is my last slide. I leave you with this conceptual framework again in trying to deliberately think about what are we missing when we are taking a statistical approach or a modeling approach or a basic analysis of whatever the in vivo is telling us, but rather, is there something fundamentally new knowledge that can be learned when we put them all together in ways that there is no way we can see even a partial answer by opening these other windows. The window analogy is actually incorrect because window analogy says it's giving you a partial answer. It's not. You're getting potentially an incorrect answer. Until you put it together, you don't know what the actual reality possibly is. So with that, I'll stop here, and I guess we'll take questions at the panel time. Thank you.